Hello viewers, this is Dr. P. Mary Anupama with the second video of nitrogen fixation, which talks about the types of nitrogen fixation. Nitrogen fixation, it's of two types. We have abiotic nitrogen fixation, or which is the atmospheric nitrogen fixation, and then the biotic types. First let us talk about atmospheric fixation or the abiotic fixation. Only 5 to 8% of the atmospheric nitrogen it is fixed by this abiotic or atmospheric fixation. It is due to lightning. This lightning it causes splitting of the nitrogen molecule, which in turn combines with oxygen to form nitrogen oxide. These nitrogen oxides, as it is raining, they dissolve in the rain and then they form the nitrates and the nitrites, which are carried to the soil. So you see here the nitrogen is combining with the oxygen, then you have the nitrogen oxides formation followed by that dissolution in the rain, which change them into nitrates, and then as they reach the soil these become available for the plants. So this is how the nitrogen bonds are broken, and finally you have nitrate formation, and this nitrate is now available for the plants for their consumption, and then they immobilize the plants, they immobilize the nitrates into these amino acids and then into the proteins, which is then transferred to various levels of the food chain. Next is our biological nitrogen fixation. This biological nitrogen fixation, it was initially discovered by the German agronomist Hermann Hellriegel and the Dutch microbiologist Martinus Beijerinck. So this biological nitrogen fixation, it occurs when the atmospheric nitrogen it is converted to ammonia by the enzyme nitrogenase, which is present in the living organisms. And this biological nitrogen fixation, it is the very much alternative, very valuable alternative to nitrogen fertilizers. You do not use or give nitrogen fertilizers, rather you can depend on biological nitrogen fixation, and this we call it as the organic farming nowadays. It is carried out very much naturally by the microorganisms which are present there, and these we term them as diazotrophs, and these include bacteria like azotobacter and even archaebacteria. All biological nitrogen fixation, it is mainly carried out by the enzyme nitrogenases. These enzymes they contain iron, often again they have one more element, that is molybdenum, and sometimes they even have vanadium. So the ecology of nitrogen fixing bacteria. So once this is the system of nitrogen fixation wherein nitrogen is converted to ammonia, and here the energy source, that is the carbon source, that is used during these various types of nitrogen fixation, and then the estimate of nitrogen fixation, that means how much of nitrogen is fixed per hectare per year, is represented here. We have three types of biological nitrogen fixation, like symbiosis can be taking place, association, and even nitrogen fixation by free living microbes. So first is symbiosis, which all of us know, which is done by rhizobium. These rhizobium they require sucrose as the carbon source, and this they get it from the plants, the host plants, and annually you see that it is around 50 to 400 kilograms of nitrogen is fixed by symbiosis. Next is association by azospirillum. Example is azospirillum. So here the roots exudates of these host plants, whatever they give out, they become the organic carbon source for this azospirillum, and annually you see 10 kgs to 200 kgs of nitrogen is fixed per hectare by azospirillum. Next one is the free living bacterium, that is rhodospirillum, which you see around in the soil. So these you find them around the plants, and you have this heterotrophic plant residues, or even there some of these can be photosynthetic. Okay, these can be autotrophs, that is photosynthetic. So this heterotroph type of living organisms or the microbes, they produce around 1 to 2 kilograms of nitrogen annually, while the autotrophs they produce around 10 to 80 kilograms of nitrogen per hectare per year. So coming to this diagrammatic representation of the primary nitrogen fixing genera of the microorganisms, these we call them as diisotrophs. Diisotrophs again can be categorized into two types. We have the heterotrophs and then the photoautotrophs. Among the heterotrophs, you have free living as well as symbiotic. So among the free living, again we have aerobic bacteria, facultative anaerobes, and then anaerobes. Under aerobic, you have all these types of organisms that includes Azotobacter, Azomonas, Bergenicia, Dergsia, Rhizobium, and Azospirillum. Next, among the facultative anaerobes, you have Bacillus and Klebsiella. And among the anaerobes, you have Clostridium, Desulfovibrio, Desulfotomaculum, and Methanobacterium. Coming to the symbiotic ones, again two types, either they can form nodules or they need not form nodules. So you have non-nodule forming as well as nodule forming bacteria. Non-nodule forming include the rhizosphere, that is rhizosphere around the area of the roots, they will be there. Okay. Under rhizosphere, we have the examples Azotobacter and Azospirillum. Then we have phyllosphere. We have examples again Azotobacter, Bergenicia, Klebsiella. Then coming to the nodule formation, we have two categories, legume and non-legume. The famous one which we'll be studying is the Rhizobium one, and the non-legume, it includes Frankia, Rhizobium, nostoc, etc. Now coming to the photoautotrophs as nitrogen fixers, again these are of two types, the free living and symbiotic. Among free living we have cyanobacteria and other bacteria. Cyanobacteria include Anabaena, Calothrix, Glyocapsa, nostoc, Trichodesmium. Other bacteria include Chlorobium, Chromatium, Rhodopseudomonas, Rhodospirillum. Coming to the symbiotic ones, again we have non-nodule forming as well as nodule forming. Non-nodule forming include Anabaena, Holosyphon, nostoc, and nodule forming include nostoc. Symbiotic nitrogen fixation. This is the most important one. Symbiotic nitrogen fixation, we see this in the legume plants, wherein the bacteria, they reside in the root nodules of these plants, that is the legume plants. These bacteria, they belong to the genus Rhizobium, and they can fix the atmospheric nitrogen when they are in association with these leguminaceae plants. And finally, they form these nodules in the roots. The bacteria, they accumulate initially within the vicinity due to some factors, growth factors, which are released by the roots of these plants, and then they penetrate into the roots of these plants, that is the root hairs, and then they form the infection thread. So once the infection thread is formed, it goes to the cortex and then it reaches into the endodermis or even the pericycle of the host roots, and then there the bacteria, they start replicating. And what happens is, during this process, the host cells, they also start dividing, they double their chromosomal count, and then they start forming nodules. So successful nodulation takes place only when the bacteria, they're able to invade the cells with that double count of uh, chromosomes, that is with the double the normal somatic complement chromosomes, it should be double. So this is the root hair, this is the soil vicinity, and here we have represented the epidermis. Here you have the cortical parenchymal cells, and the bacteria will be in the soil. So as this rhizobia, as they invade or as they come close to this thing, I shall tell the details later. So what happens is, initially you can see this curvature of the root tip, and then the infection thread will be formed, and then this infection thread goes down to the cortical parenchymal cells, it reaches the meristematic cells, and then nodulation is initiated. So here let us see what happens. See, some books have told that it is lectins also that contributes to this nodulation in some of the plants. So the root hairs of these plants, they secrete some lectins. Some of the legumes, they secrete lectins. and this is the bacterium. It has the surface polysaccharide. And what you see is that when there is an interaction between the surface polysaccharide and the legume lectins, okay, this interaction is again host specific. Now initially this two, since they are specific, they come together, and then you can see that the rhizobium get attached to the root hair of the host legume. So once attached, then the curvature of the root hair will be taking place. Meanwhile, here you see that this mitotic area where you have the meristematic cells, they are stimulated and they start doubling their chromosomal count. The infection thread goes there, and then finally you see that the infection thread reaches there and tetraploid cells are formed, and this is the young nodule representation over here. Now let us see the exact schematic portrayal of this interaction between legume roots and rhizobium. Please see this. You see that the rhizobia, they have the symplasmids which code for nod D. Okay, nod D. This nod D protein, it is present on the surface, okay, the bacterial surface. And here the root hairs of these legume plants, they give out some flavonoids, and these flavonoids, these as they reach the rhizobium bacteria, you see that NOD activation is taking place because of the interaction between the flavonoid and the NOD. Now you can see here the NOD, it binds to the NOD box which is present in the promoter region, okay, that induces the NOD genes, the promoter of the NOD genes, and then it induces the production of these proteins which you call them as the NOD factors. So here you can see as this is bound here, the NOD, you see that the NOD factor synthesis is initiated and you have the NOD proteins, the NOD factors which are released. These NOD factors which are secreted by the rhizobium, okay, they are attached to the NOD factor receptor on the plant root hairs. The plant root hairs have NOD factor receptor. So these go and bind there, and these induce the root hair deformation and then the nodule formation in the cortex. Okay, so these are the main important steps of this infection process. So once again, this is the rhizobium, and what happens is the root hair curling is taking place because of the infection thread. The branches of the infection thread, they penetrate the cortex, okay, and the cells, they start proliferating, and then you have the nodule, root nodule formation which is taking place here. Okay, so these root nodules, we have seen that they are red in color. They consist of this pigment like hemoglobin, and it appears like it's a product of rhizobium and then legume complex. And this is the structure of the leg hemoglobin. And the rate of nitrogen fixation, it increases with increase in leg hemoglobin content. That means the more of leg hemoglobin, the more of nitrogen fixation that is taking place. In non-leguminaceous plants, the nitrogen fixation can be symbiotic and nodules that have actinomycetes. We also have this asymbiotic nitrogen fixation. You have several bacteria which are doing it, and there are various processes of asymbiotic nitrogen fixation. That means they don't associate with anything. So we have anaerobic bacterium like Clostridium which does it, aerobic bacterium like Azotobacter bacillus, Chlorobium, Chromatium, Methanobacterium, Pseudomonas, Perillum, etc. Next, you also have heterocystus blue-green algae which do it, like the Anabina, Anabenopsis, Olocera, Calothrix, Chlorogloia, Cylindrospermum, Fischella, Hippelosiphon, Mastigocladus, Nostoc, Syntonema, Stigonema, Tolipothrix, and Vestulopsis, etc. So these are some images of various organisms that I could download. This is Aponotheca, Meristomopoda, Chlorococcus. This is our Oscillatoria, and here you have our Anibina, Nostoc, Gliotricha, Stygonema, Tolipothrix, Rivieraria, some nitrogen fixing ones. Now let us focus on the major general requirements for effective nitrogen fixation to take place. First important one is there should be a protective mechanism for nitrogenase against oxygen. The nitrogenase is very sensitive to oxygen. Next, you should have a source for hydrogen as well as good energy source, that is ATP source should be there. This nitrogenase, it should be in active form. Next, you also need a carbon compound like keto acid, that is alpha ketoglutaric acid or glutaric acid, so which can take up ammonia. Say for example alpha ketoglutaric acid, it can take up ammonia and get converted into glutaric acid, while this glutamic acid, and this glutamic acid can take up ammonia to form glutamine. So like that you should have some carbon compound which can take up the ammonia. Next, hydrogen, it is used for the conversion of this nitrogen to ammonia, and then this takes place with the help of ATP. And here you require both hydrogen as well as ATP, and these come from either photochemical reaction of photosynthesis or even from aerobic respiration. Next, you also need reduced pyridoxine or flavoproteins which act as reductants. All these 8 are the requirements for nitrogen fixation to take place. One more essential requirement here is the minerals molybdenum, iron and calcium for nitrogen fixation to take place. So let us see how the electron energy transfer takes place in nitrogen fixation. So this is the nitrogen fixation wherein you have the nitrogenase which converts the nitrogen to ammonia. And here we have talked about the requirement for ATP, the energy source, hydrogen, pyridoxine. So the pyruvate is the source for acetyl phosphate as well as the reducing parts, which in turn convert the pyridoxine. Pyridoxine, it takes up the protons and then transfers them to nitrogen to convert it to ammonia. While the energy it comes from this acetyl phosphate. This acetyl phosphate, it splits up into acetate and the phosphorus is transferred to ADP to form ATP. So this ATP is the energy source that is involved in splitting off the triple bond of nitrogen and converting it into, reducing it into ammonia. This is the overall equation of biological nitrogen fixation. So you see that it is extremely energy consuming, and this is because of majorly the triple bond that exists in the nitrogen. So you can see here, to convert one nitrogen molecule to 2 molecules of ammonia, it requires 8 pyridoxine molecules, 8 protons, 16 mg ATP, 18 water molecules. Then you get 2 ammonia molecules, 2 OH groups, then the oxidized one, pyridoxine, and you get 16 mg ADP, because ATP, it has donated the phosphate, phosphorus is broken, phosphate bond, so you get ADP plus phosphoric acid from this, and then some hydrogen. So this is the overall equation of biological nitrogen fixation. We have to understand about the enzymology of this. It is a nitrogenase and it requires the reductants, that is the pyridoxine, that is the reduced pyridoxine, ATP and oxygen free conditions as we have told, and you have the regulatory controls, that is if ADP is more, it inhibits uh, ammonia formation, and even if ammonia is more, ammonia, that it is the end product, it inhibits expression of NIF genes, so you don't get nitrogenase. So all these conditions are required. So what is exactly biological nitrogen fixation is? It's nothing but the reduction of atmospheric nitrogen to ammonia. And this is done by our oxygen sensitive enzyme nitrogenase. And we are talking about the reducing parts which are required, that is NADPH or ferredoxin via iron molybdenum center. Via iron molybdenum center. And here one more point is the plant genomes, they don't have any genes that code for this nitrogenase. Only prokaryotes have it. So plants have to depend on these microbes. And even in the bacteria, only certain free living bacteria like Klebsiella, Azospirillum, Azotobacter, some blue-green algae, and few symbiotic rhizobial species, they can fix this nitrogen. Another nitrogen fixation phenomena that exists is with the actinomycetes and even this alder species, that is alnus species. So the nitrogen majorly it is converted to ammonia by the nitrogenase. It catalyzes the conversion of dinitrogen into ammonia as shown in this equation. Okay. So the above reaction, it seems very simple, and the entire atmospheric nitrogen, it can be easily converted. Can it be easily converted entirely into ammonia? So why is this particular enzyme study more important? You can see here that the nitrogen, the two atoms of nitrogen, they are linked by triple bond. And this triple bond breaking it becomes very, very difficult, And nitrogenase, we have seen that it consumes 16 moles of ATP for every molecule of nitrogen, and then only it is reduced to ammonia. So this is a very expensive, energy expensive process in the nature. So that's why all the nitrogen you cannot expect to be converted into ammonia just like that. And this nitrogenase complex, it consists of two proteins, that is nitrogenase reductase and then nitrogenase. As such, the nitrogenase enzyme is a big complex. What does it consist of? It consists of two protein components. One is the nitrogenase reductase and then the nitrogenase. This nitrogenase reductase, it's a 60 kilodalton homodimer with a single iron sulfur cluster. 4 iron and 4 sulfur molecules will be there. And it's very sensitive to oxygen. It binds magnesium ATP, and you have 4 ATP, they are required per pair of electron to be transferred. Per pair of electron to be transferred. And reduction of nitrogen to ammonia plus H2. The end product is ammonia plus H2. It requires 4 pairs of electrons, 16 ATP. They are consumed, okay, per nitrogen, as we have seen previously. And as I told you, the triple bond has to be broken and then ammonia has to be formed. So there is a activation barrier, very high activation energy is required. That's why it requires lots of ATP, 16 ATP to cross this activation barrier. So here we'll explain this. Why should nitrogenase need ATP? Yes, nitrogen reduction to ammonia, it is thermodynamically favorable. However, there is a barrier, that is the triple bond has to be broken, which requires that 16 ATP. That is it provides the activation energy that is required for this conversion. Next, this coming to this nitrogenase, the second component, it is a 220 kilodalton heterotetramer, and each molecule of this nitrogenase, it consists of 2 molybdenums, 32 iron and 30 equivalents of acid labile sulfur, that is in the form of iron sulfur clusters. There are 30 iron sulfur clusters in this, and 4 iron sulfur clusters plus 2 iron molybdenum cobalt and an iron molybdenum cofactor. So nitrogenase is low, that is 12 electron, it takes up only 12 electron pairs per second. So only 3 molecules of nitrogen per second can be processed by nitrogenase. So here is a representation of the mechanism of nitrogen reduction up to the level of ammonia. We have seen that there is reduced ferredoxin and then ATP. Reduced ferredoxin provides hydrogens. So the triple bond and the nitrogenase enzyme, initially it is converted, it is reduced to NH, NH linked by a double bond, then NH2, NH2 linked by a single bond, which is further broken, and you have ammonia and nitrogenase. So this is how systematically the nitrogenase enzyme converts nitrogen to ammonia. So here you have the pyruvate hydrogen NAD, they reduce the ferredoxin, and you have the nitrogenase reductase. Okay, this nitrogenase reductase, it consumes the energy, and then the nitrogenase comes into action, which takes up the electrons which are released and the protons, and finally converts the nitrogen into ammonia. We have this various categories of nitrogen fixations. Already we have seen about non-symbiotic ones. You also have associative and symbiotic ones. Symbiotic we have studied. So non-symbiotic or asymbiotic, that is by the free living organisms, that is the blue-green algae, anaerobic bacteria. We call them as the non-symbiotic biological nitrogen fixation process. And already in the classification we have seen photosynthetic and non-photosynthetic. Photosynthetic example is Chlorobium chromaceum. Non-photosynthetic example is Azotobacter, Azomonas, Dergsia, Beijeringia. We have seen this previously. Just again a recall. Once again a recall of free living and aerobic nitrogen fixers, photosynthetic and non-photosynthetic. Photosynthetic is Rhodospirillum, non-photosynthetic is Clostridium. Among the free living chemosynthetic bacteria, you have this heterotrophic one, Desulfovibrio. And cyanobacteria, we have we'll be seeing one more topic as a continuation of this. There you'll be studying about this heterosis bearing nitrogen bearing algae which do this nitrogen fixation, that is photosynthetic nitrogen fixation. So heterosis bearing ones like Nostoc, Anabena, Rivularia, Calothrix, and non-heterosis bearing ones are Oscillatoria, Gloeocapsa, Lynchbia, and Plectonema. In free living fungi, we say yeast and Pullaria duet. So the symbiotic free living ones, these are considered to be the primitive ones, and the reduction process in them, it's independent of respiration. So these organisms, they just fix the nitrogen more actively under even under poor aeration. That means if less air is there, they do it very nicely, provided that there is no hydrogen gas that is being produced. Now let us talk about this associative symbiotic nitrogen fixation. So there are certain bacteria which live in close vicinity of the roots of some plants of cereals and some grasses, and then they fix the nitrogen also. So this association, you say it as it is loosely mutualism, and you call it as associative symbiosis. So these bacteria, they reside in that transition, like in the zone of the rhizosphere, in between the roots and the soil, that is called the rhizosphere. And at times they even enter the roots also. Some of the fixed nitrogen, it is absorbed by the roots of these plants um, through the root hairs, and in turn the bacteria, what they get is they get the nourishment, that is the carbohydrate supplements from the roots, which are the roots start releasing some carbohydrates, so they consume it. Okay, some examples are the Azospirillum brasiliensis, it is found in association with the cereal roots. You have Beijeringia, found in association with the roots of sugarcane. You have Azotobacter paspali. It is found in association with the roots of tropical grass like Paspalum notatum. Next, nitrogen fixation in photosynthetic bacteria. So we know that cyanobacteria, these are oxygen emitting photosynthetic bacteria, and you find them in ocean, even in the fresh water, and even the terrestrial environment they're present, everywhere, and they are also capable of fixing the nitrogen. But you see that we have seen previously, nitrogen is this enzyme which is responsible for the reduction of nitrogen. It is very much sensitive to oxygen. So therefore, this oxygenic photosynthesis and nitrogen fixation, they are not that compatible. So oxygen synthesis, that is photosynthesis, and nitrogen fixation, because photosynthesis releases oxygen and oxygen inhibits nitrogen fixation. So they both are not compatible. And these cyanobacteria, they are so smart that they have come up with a strategy which avoids these situations, allows the plant to do photosynthesis as well as they produce nitrogen. Okay, it allows them to fix the nitrogen also. So some filamentous cyanobacteria, they are there. Like if both are there, they cannot do it, right? So they have come up with some differentiative heterosis. So heterosis, these are specialized cells that lack this photosynthesis phenomena. That means they don't have the photosynthetic apparatus. Rather, they have some glycolipid cell wall in them, and this glycolipid cell wall, it helps them, it helps them to stay away from the oxygen. So that means it maintains, because of these glycolipids, it maintains low amounts of oxygen so that nitrogen fixation can take place very effectively. So this strategy, which is adopted by this filamentous cyanobacteria having this heterosis, you call it as spatial separation. Spatial separation of what? Spatial separation of this oxygenic photosynthesis as well as nitrogen fixation. There are also non-heterocystous cyanobacteria. They temporarily separate this process by nitrogen fixing. It takes place during the night, while in the daytime they do the photosynthesis. So there is a time gap. They do that in heterocystous ones. In filamentous ones having heterosis, heterosis are specialized ones wherein they lack photosynthesis apparatus. While in non-heterocystous ones, photosynthesis takes place in the daytime, while nitrogen fixation takes place in the night time. So cyanobacteria, they fix this carbon dioxide and also nitrogen. So these two things will be taking place. So fixing of nitrogen, again we know that it's a high energy requirement process because of the ATP. And cyanobacteria, they are phototrophic organisms, so they use sunlight and they convert it into energy that meets the demand of nitrogen fixation. So this nitrogen is it's complex, and we have seen that it is sensitive to nitrogen and requires anoxic environment, that means less oxygen. So cyanobacteria, these are phototrophic organisms, and these are the strategies which they have come up, that is spatial separation. We call it as spatial separation. Already I have told about this in the previous one. So nitrogen fixation in the ocean, it is restricted to subtropics, and it is carried out by the free living non-heterocystous ones. Non-heterocystous means they carry out photosynthesis in the daytime and nitrogen fixation in the night time. So you have unicellular cyanobacteria, symbiotic cyanobacteria living with microalgae, all such things, they are doing this process. So you see this heterocystous ones, we find them in the freshwater, brackish water, and even in the terrestrial ones. And the cyanobacteria, they maintain this circadian clock that synchronizes the process, and allows them to fix the nitrogen effectively. So here you can see the heterocyst. Okay, these are the algae wherein you can see heterocyst, filamentous algae. So this is the normal one, vegetative cell, wherein you have the photosynthesis taking place. So carbon dioxide fixation has taken place, and during this process oxygen is released also, ATP is generated, and along with that we have seen there is a requirement of one acceptor for ammonia. So here it is the glutamine which is the acceptor for ammonia. Glutamine comes from oxaloacetate to alpha ketoglutarate and then to glutamate, and this glutamine, it takes up the ammonia. Okay, it takes up the ammonia and again it gets back here. Okay, glutamate takes up the glutamine and it gets back here. And here you see the nitrogen fixation which has taken place. The nitrogen is converted into ammonia when less oxygen is there. Okay, here respiration is taking place, lots of respiration is taking place. So ATP is produced, and ATP meets the energy requirement for breaking the triple bond here. This is the heterocyst. This is the heterocyst wherein ammonia is produced. Ammonia can also go into the outside environment, and even the glutamine can diffuse out and it can reach the plants. The plants can take these things. Okay, and here the plants, they produce some carbohydrates, right? So that sucrose is also taken up by this filamentous algae. And you know that sucrose can be broken into glucose and fructose, and then glucose enters the glycolysis. All this will be taking place. Okay, and then energy will be released, and then again you see glutamate production. Glutamate again enters here to take up ammonia, and then glutamine is formed. So this is in the case of filamentous algae having heterosis. And here I have to talk about this high oxygen stress. So when high oxygen stress is there, actually it causes proteolysis of the nitrogenase subunits. So what you see is that the nitrogenase synthesis, it is suppressed, and thereby it leads to shortage of those respiratory substrates and the reductants, and the necessary nitrogen fixation and assimilation, it is inhibited. So inhibitory effects of moderate levels or short exposure times in vivo, it is reversed, leading to increase in nitrogen fixation. So if moderate levels of oxygen are there, then it's okay. And some diazotrophs, post-translational modification of iron protein from an inactive to active form, that has to be done. Even this is important, inhibitory effects will be there if such things are not there properly, if oxygen levels are not maintained properly. And these diazotrophic cyanobacteria, they provide this bulk nitrogen in the surface of the oceans, in oceans. So diazotrophs, they actively produce oxygen by photosynthesis, and during the daytime they do this. So in the daytime, nitrogen production is restricted. So you see that this is the vegetative cell or the heterocyst, wherein you can see the glucose, it's metabolized into glycogen, and you have this reduction groups which are produced. These reduction groups, they contribute to the reduction of this iron sulfur proteins, which in turn are required by the nitrogenase for conversion of nitrogen to ammonia. This ammonia is taken up by glutamate, glutamic acid, and then glutamine is produced, okay, in the heterosis. So all these will be taking place in the heterosis. So here is a small comparison between free living nitrogen fixers and symbiotic nitrogen fixers. So symbiotic nitrogen fixation, it is performed by some bacteria like rhizobia or Franknia, and the free living nitrogen fixers, what they do is like you have this dissolved organic carbon which exists in the soil, or some complex carbon sources, whatever they say, they are there. While symbiotic nitrogen fixers, here they have to consume the whatever that is available here, while in the case of symbiotic nitrogen fixers, they are supplied with this carbon source. Okay, they are supplied with the carbon source. Sucrose will be there, or succinate is provided by this plants, host plants, initially sucrose and then succinate. The oxygen concentration in the rhizosphere, it is highly variable, and it depends upon the soil's the structure or the texture. Say for example clay soils, they are quite compact, so less oxygen will be there. But if it's a sandy soil or some pore size is there, some red soils and all, the texture it varies, and it decides the oxygen concentration within the soil. So symbiotic nitrogen fixers, they're supplied with oxygen at very low concentration by the plants itself, because they are present in the root nodules. While here, while these free living ones, they may be diazotrophs. They require phosphorus, iron, molybdenum, vanadium, nutrients and all, they are to be supplied. And presence of oxygen, that is again a hindering, it is hindering. So oxygen supply will not be there for these things. Whatever that is there in the soil, they take it. Okay, it comes from the soil itself. However, the nutrients, they are delivered to the symbiotic nitrogen fixers by the plants, but here it's nothing like that. Diazotrophs in the rhizosphere, they can access nitrogen from the soil and even the free living microorganisms. While all symbiotically fixed nitrogen, whatever that is fixed here, it goes directly to the plants. While here, whatever they fix, it has to diffuse to the soil, the microorganisms, the plants, whatever that are existing there, they can take. While here, it supplies directly to the plants. Okay, so based upon the habitat, the free living and the symbiotic nitrogen fixers, based upon their habitat, their level of fixing varies. So here they enjoy the supplies and it's symbiotic mutual association. They enjoy the supplies and they supply the plants with nitrogen. While here they have to struggle for the carbons, whatever that are there, they have to take them, and then they give out the they take the whatever carbon that is present, organic carbon that is present in the soil, and then they give out the nitrogen. So whatever in the rhizosphere the plants are there, the plant roots, if it is in the rhizosphere, they consume it happily. And even the other ones also, they can take this fixed nitrogen. So this is end of the part 2 of nitrogen fixation. Thank you.